Thank you for the introduction. First, I'd like to thank the organizers for giving me this opportunity. Today, I'd like to talk about the function of protection of telomere 1A, POT1A, in the regulation of the self-renewal of hematopoietic stem cells. As you know, the end of linear chromosome have the potential that can activate ATM and ATR-dependent DNA damage response, DDR. Because this region contains the constitutive single-strand DNA and DNA ends. Therefore, we need a protection mechanism for the prevention of unnecessary DNA damage response, DDR, at telomeres. In this regard, telomere-specific protein complex sheltering and T-loop structure contribute to the prevention of the telomeric DDR. The sheltering is composed of 6 protein components: TRF1, TRF2, LAP1, TIN2, TPP1, and POT1. And this protein complex is necessary for the prevention of the DNA damage response signaling, double-strand break repair, and 5' end resection. This slide shows the function of POT1. It has been reported that human sheltering contains single POT1 protein, but mouse sheltering contains 2 POT1, POT1A and POT1B, which have a different function at telomeres. And POT1 specifically binds to the single-strand DNA via OB4 domains and prevent the binding of replication protein A, RPA, to this single-strand DNA, then inhibit ATR-dependent DDR signaling. And this protein also contribute to the control of the 3' overhang length and telomere length. And importantly, this POT1 is necessary for the repression of the sister telomere fusion. Then in this study, we analyzed the function of POT1A in the regulation of the hematopoietic stem cells, HSCs. First, we analyzed the expression of POT1A in the hematopoietic cells. As you can see, POT1 is highly expressed in the long-term HSCs and short-term HSCs compared with the progenitor cells and differentiated cells. Then next, we analyzed the function of POT1A. First, we transduced the POT1A specific shRNA into the hematopoietic stem progenitor cells and transplanted into the lethally irradiated recipient mice. Then we analyzed the long-term engraftment of the donor HSCs. As you can see, transduction of the SH-POT1A significantly reduced the long-term reconstitution of the donor cells. This data suggests that POT1A is involved in the self-renewal of the HSCs. Next, we transduced the POT1, overexpressed the POT1A in HSCs and performed the serial bone marrow transplantation to evaluate the function of POT1A in the self-renewal activity of the HSCs. As you can see, after this first BMT, there is no significant difference in the reconstitution between POT1A and control GFP overexpressing cells. After 4 months of first bone marrow transplantation, we isolated the GFP positive donor HSCs and transplanted into the secondary recipient mice. As you can see, the cells overexpressing POT1A shows the higher reconstitution compared to the control GFP transduced HSCs. This data suggests that overexpression of POT1A enhanced the self-renewal activity of HSCs. Then, to investigate the function of POT1, how POT1A enhanced the self-renewal of HSCs, we focused on the inhibition of the telomeric DDR in HSCs. This slide shows the telomeric DDR in HSCs. The green dots are TRF1, which is a marker for the telomeric DNA, and red color indicates 53BP1, which is a marker for the DNA damage. Then telomeric DDR, telomere dysfunction-induced foci are identified the co-localization of the TRF1 and 53BP1. Then first we analyze the effect of the knockdown of POT1A in the telomeric DDR in HSCs. As you can see, knockdown of POT1A significantly increased the number of the TIF in HSC after the culture. Then next we examine the effect of the overexpression of POT1A on the inhibition of the TIF formation. We transduced the POT1A into the HSC and transplanted into the recipient mice. After the 4 months of the bone marrow transplantation, we isolated the GFP-positive donor-derived HSCs. As you can see, the overexpression of POT1A significantly reduced the number of TIFs. This data suggests that POT1A controls the self-renewal activity of HSC through the inhibition of the telomeric DDR in HSCs. Then, from these findings, we saw that POT1 is the potent tool for the ex vivo expansion of HSCs. Then, to use the POT1A for the ex vivo expansion of HSC, we produced the soluble recombinant POT1A protein, which has the his tag and MTM tag. And this MTM is a membrane translocating motif composed of 12 amino acids from hydrophobic signal sequence from FGF4. And this MTM tag mediates the uptake protein into the cells. By using this method, we can transduce the POT1A protein into the HSC without virus transductions, and we can control the concentration of the protein. And more importantly, this method is very easy and very efficient method for the transduction of the protein into the primary HSCs. Here I shows the efficiency of the transduction of the POT1A. As you can see, most of HSC incorporated MTM POT1A protein within 2 hours, and incorporated protein was detectable in HSC at least 96 hours. And more importantly, incorporated MTM POT1A, the co-localization of the MTM POT1A and TRF1 shows that incorporated proteins are located in telomeric region in HSCs. Then we analyze the effect of MTM POT1A on the prevention of telomeric DDR in HSCs. We cultured long-term HSC with or without MTM POT1A for 3 weeks. Then we analyzed the telomeric DDR in cultured long-term HSCs. As you can see, the treatment with MTM POT1A significantly reduced the number of TIFs. Then to investigate further the function of MTM POT1A on the maintenance of the HSCs, we analyzed the individual HSC cell division patterns. Then we cultured long-term HSC with MTM POT1A or control MTM protein for 10 days. Then after 10 days of the culture, we analyzed the cell division pattern of the long-term HSCs. As you can see, treatment with MTM POT1A significantly increased the HSC expanding symmetric stem cell division and decreased the progenitor expanding, symmetric progenitor division in cultured HSCs. Then the number of HSCs are increased in cultures. Then next we analyze the number of long-term HSCs after the culture. We cultured long-term HSC with MTM POT1A for 3 weeks, then evaluated the number of the long-term HSCs. As you can see, the treatment with MTM POT1A increased the number of LSK CD41 negative, CD48 negative, CD150 positive long-term HSC after the culture. Then we evaluated the number of the functional long-term HSC by the limiting dilution competitive bone marrow transplantation using 10 days cultured long-term HSCs. As you can see, MTM POT1A induced a 3-fold enrichment of long-term HSCs after the culture. We also analyzed the function of MTM POT1A in the maintenance of the self-renewal activity of HSCs. We cultured long-term HSC for 10 days with MTM POT1A and performed the serial bone marrow transplantation. As you can see, the cells cultured with MTM POT1A shows the higher long-term reconstitution after the serial bone marrow transplantation. These data suggest that MTM POT1A maintained or enhanced the self-renewal activity of HSC in culture. Then next, we analyze the effect of human POT1 on the expansion of human HSCs. To analyze the function of human POT1 in the regulation of human HSC, we prepared MTM human POT1 protein and cultured cord blood-derived long-term HSC for 10 days with or without MTM human POT1 protein. As you can see, similar to the mice MTM POT1A in mice HSCs, MTM human POT1 significantly reduced the number of TIFs in cultured human HSCs. And we also observed that MTM human POT1 increased the number of human long-term HSC in cultures. Finally, we analyzed the number of functional human long-term HSC after the culture using the limiting dilution bone marrow transplantation. And we observed that MTM POT1 increased the 4.5-fold, induced a 4.5-fold increase of the long-term HSCs compared to the control proteins. This is a summary of my presentation. In this study, we found that HSC expressed high level of POT1A. A knockdown of POT1A increased the telomeric DDR in HSC and reduced long-term reconstitution activity of HSCs. An overexpression of POT1A or treatment with MTM POT1A or human POT1 prevented telomeric DDR in HSCs and maintained HSC self-renewal activity. And we propose that POT1 will be significant use in the development of robust and safe method for ex vivo expansion of human HSCs. Finally, I would like to thank the people who contribute to this work. Thank you for your attention.